So welcome. This is kind of an experimental program that we're doing here in the clinic. And before we dive into what is probably the more fun stuff that y'all are looking for, we thought it would be a good idea to give you, not knowing how many of you are familiar with the Plant and Insect Diagnostic Clinic here at Iowa State University. We wanted to give you a little bit of a background of who we are and what we do. So, the Iowa State University Plant and Insect Diagnostic Clinic, or PIDC, is staffed by two diagnosticians, Zach and myself. I am the plant disease diagnostician, and Zach is our insect diagnostician. We have both been here since October of 22. So we just, both of us just passed our 2-year mark here at Iowa State. Yeah, I think we've really been enjoying our jobs here and working together. The Plant and Insect Diagnostic Clinic's mission is to provide accurate and timely plant health diagnoses to our clients. So this is mostly with the physical samples that we receive in the clinic, although often we appreciate communicating with clients and seeing pictures prior to receiving samples. We also, as we complete our diagnoses, we write up sample reports for each of our clients. That includes information about what we found in their sample and how to manage it, especially with integrated pest management tactics in mind. The services that we provide are available to anyone, homeowners, farmers, extension, commercial service providers. And it's not just for the state of Iowa. We do accept samples from any state within the continental US, as long as we don't get soil from a soil quarantine area in the southeast. Some of the services we provide, what I do is the plant problem diagnostics. This includes basic microscopy, isolation and culturing from plant tissue and moist chamber, which is a process to help increase or induce sporulation of fungal pathogens. There are some optional add-ons to that service, doing either molecular or serological assays. And then we also offer insect identification. We're one of two labs in the United States that offer molecular insect identification, which is a service that Zach brought here with him from Utah. And then we do a lot of soybean cyst nematode egg counts and complete nematode counts from soil samples. And it's important for us to set your expectations in a realistic place so you know what we can do and what we can't do. We do plant problem diagnostics, either whole or plant parts. We will do insect, spider, tick, and mite identification, and we can do plant parasitic nematode analyses. What we don't do that some people think we do not do mushroom, weed, or plant identification. Mushrooms, that's just for liability reasons. Our county offices can help sometimes with plant identification and some of the online resources that we have. We do not do soil or plant nutrient analyses, but we do have a website that we can share with folks that has a list of laboratories that offer these services. And we also don't do herbicide residue testing. And we have a document that we can share with folks if they're interested in getting their plant tissue tested for herbicide residue. Some of the diagnostic methods we use in our diagnostic lab include microscopy. This is both compound microscopy, which these spores here are an example of what I might see under the compound microscope, getting a close-up look at spores and the morphology of the fungi associated with plants. But we usually start with microscopy under the dissecting microscope, where I'm actually able to look at the fungal structures that form on plant parts, such as this right here is an example of stigmina, of needle disease of spruce trees. We do culturing and isolation. This is for both fungal pathogens, which this is an example of here, and bacterial pathogens, which is the example on the right-hand side there. Then we do molecular assays and also have the tools to do serological and lateral flow device assays. This is an example of what a lateral flow assay looks like. They're for specific pathogens, and they're basically like COVID tests, where if we have two lines on the test, that means it's positive. So in this case, this sample was positive for potyvirus, which is a group of viruses that cause disease on plants. And then we do nematode extractions. Most of the nematode extractions we're doing are from soil, but there are some nematodes that can cause issues in garden plants, or in this case, pine trees, and those nematode extractions look a little bit different. We have some different diagnostic categories, and this will be important to keep in mind as we get into some of the numbers from this year. But basically, every time we make a diagnosis, before we send it out to the clients, we select at least one of these categories, depending on what we find with the sample. So either we found a pathogen associated with the sample, we found an arthropod or insect associated with that sample, or we didn't find any pathogens or arthropods, so we suspect an abiotic issue. Other is kind of a catch-all category that we seldom use because it doesn't really make sense to us. And then we added this year the category non-validated sample type. And this is for the samples that we get that are not appropriate for the diagnostics we're doing, but the client still would like us to do, check for plant diseases or insect pests. We are the National Plant Diagnostic Network Laboratory for the state of Iowa. The National Plant Diagnostic Network spans all of the United States, including its territories. So each of these states has at least one NPDN lab. For Iowa, we are the only NPDN lab. What am I doing? Okay. So the National Plant Diagnostic Network was formed in 2002 following 9/11 as a response to try and unite and build a way for these diagnostic labs across the country that already existed, build a way for us to communicate, and now it serves as a premier diagnostic system in the United States with the purposes of quickly detecting and identifying plant pests and pathogens, especially when it comes to issues of quarantine pathogens and pests in the United States. In the NPDN, we recognize that the challenges to plant health are constantly changing, and so our diagnostic systems are built to kind of reflect that and work with that in mind. And as such, as I mentioned, the NPDN is a vital part of our national biosecurity infrastructure. And that's kind of a cool thing that we get to do on the back end that not a lot of people know about, but it is an important part of what we do here as well. Now on to the fun stuff. I am going to mute myself and hand it over to Zach.
Okay, good evening again, everybody. Zach Shum here. So as Chelsea mentioned, I'm the insect diagnostician in the clinic, and I'm going to begin going over some of the trends and some of the numbers from 2024 that we received in the clinic, from samples to diagnoses. And then Chelsea and I will both jump into some general trends and types of issues we were seeing this year in 2024. Okay, so this is 1,633, the total number of samples that we received in 2024. So this encompasses all the different diagnostic services we've had. And I'll show you some year-over-year trends as well. So what 1633 looks like in comparison to some other years. So these are all of our different 2024 samples by service. So you have the service that is listed there and also color coded in the pie chart, along with the number of that particular service that we received in 2024. So out of that 1,633, Chelsea kind of alluded to this earlier. A lot of what we do are soil nematode extractions. Most of those tend to be soybean cyst nematode. So these are primarily farmers that are submitting soybean cyst nematode soil samples to us, and they will utilize those soybean cyst nematode egg counts to make planting decisions for the following year. So that's the bulk of what we do. We have ourselves and some students, undergraduate student helpers that process a lot of those samples. The next biggest service then would be plant problem diagnosis. We received 375 of those in 2024. Normally, I would say that's about normal for us, but I'll talk a little bit more about some trends related to plant problem diagnosis samples shortly. And then following that, we have all of the other services. Complete nematode counts, which probably take the longest of most of the services that we do. Insect identification, there's a really big asterisk next to the insect identification. Those 91 samples there are physical samples that we're getting in the diagnostic clinic. However, I also do a lot, hundreds of insect identifications via photo that are just emailed or sent via text message to the diagnostic clinic. Those are not reflected in these numbers. These are just physical insect samples that are sent to the lab. And insects that come in on plant samples, those are kind of looped into the plant problem diagnosis as well. So really, there are several hundred insects that are being identified reflected in these numbers. However, it's just not including all those photo diagnostics, which I would say is the majority of what I do in the clinic is photo insect identification and spider, mite, and relatives. So here we have 2024 samples by host plant type. So this is only including plant-related samples, well, and insects as well. So these are just the hosts that they are coming in on. Field crops is always the largest in the clinic, and that's primarily because of those soybean cyst nematode egg counts. So if you kind of get rid of that really big outlier of soybean cyst nematodes, there's more evenness between all the other host plant types. The biggest one we tend to see in the clinic every year are broadleaf trees. Insect ID I kind of looped in there again, so that's at 101. And then the other ones, the other types of host plants are kind of a mix. We have a little bit from coniferous trees, which is usually a larger sample set there, and then a smaller number of vegetables, perennials, and annuals. Put another asterisk on here that fruit, you'll notice, is a very small category. This is when we tend to get samples of physical fruits inside the clinic. Oftentimes we will receive trees that are bearing fruit. Honestly, most plants obviously do bear some type of fruit, but say like crab apples, oftentimes we're only testing the leaf tissue, and we will group those into things like broadleaf trees. So keep that asterisk in mind too. We do receive more than just 3 fruit-related samples every year, but a lot of them are looped into other categories. But broadleaf trees generally tends to be the largest sample or host plant type based on all the plant samples that we get. Field crops, again, is a lot of nematode samples, but also a lot of things like corn and soybean samples that come in as well. And this is depicting the diagnoses or the different types of diagnoses by all of those host plant types, only including plants here, no insects on this one, other than via diagnoses. So we have all of our pathogen diagnoses color-coded in red, abiotics, so these are, again, non-living things that we suspect are causing issues on these plants in yellow, and then insect problems that are causing some sort of damage or issue with those plants specifically. So I will note that we find many insects on a lot of plant samples, but they're only indicated here if they are actually or suspected to be causing an actual issue or ailment to that plant. And most plant samples that we get in, you can find some number of insects on them. But again, these are just arthropods that are suspected or are causing some sort of issue on that plant. So if we're looking at the diagnoses by host plant type, I'm going to start with broadleaf trees up here. And really, like, you can see that there's a mixture of pathogens, abiotic issues, and arthropods in all of these. Broadleaf trees, sorry, we're fixing an issue right here as I'm talking, so don't mind us. We see a good mixture of pathogens and abiotics on broadleaf trees, but you'll notice on a lot of these that the issues tend to be pretty dominated by abiotic conditions. This was especially true last year when we had a lot of drought conditions throughout the state. This year, we expected a lot more pathogens due to the increased moisture that we experienced in rainfall early in the spring. That doesn't necessarily mean that you're going to experience mostly abiotic conditions, more pathogen issues later in that same season. So I don't need to go through each one of these in detail. Just note that it's a mix. We do get more abiotics on most plants most years. Again, this year may have just been a little bit impacted by some of that early season rainfall. Chelsea's going to talk a little bit later about some specific diseases that were really abundant this year, suspectedly due to the excess moisture rainfall we had early in the season. There we go. And then for kind of the fun trends that I like to take a look at are diagnostic samples year over year. So these are the total number of samples that the clinic has received every year from 2018 to 2024. These numbers are generally the same most years. We see a little bit of a decline in 2022 and then a really big increase in 2023. Generally, again, we're talking about 1,633 samples here in 2024. So all of these sample numbers are between 1,200 and 2,700. But I will note that 2023 was a little bit of an outlier. We had a couple of really large clients, mostly from industry. One in particular that sent us over 1,000 plant samples. So that is why that number is very high in 2023. 2022, we're not exactly sure why those numbers were a little bit lower, but these numbers do tend to fluctuate year over year. But on average, we're somewhere in the vicinity of 1,500 samples per year in the clinic. Again, dominated mostly by soybean cyst nematode, and then plant problem diagnoses after that.
Onto the fun stuff, our trends from 2024. So starting off early in the year, this was pretty apparent that this was going to be a big problem, is apple scab. This is a fungal disease caused by the fungus Venturi inequalis, and not only did we receive a lot of samples that had apple scab this year, but I could just see it as an issue in the landscape across Iowa, and even into I'm from Northwest Illinois, so I had to tell my Mima that her crab apple was not dead, that it was probably just apple scab. So a little bit more about apple scab. For those of you who might not be familiar, again, this is a fungal disease. This disease overwinters on fallen leaves, and it can affect leaves and fruits. So the leaf symptoms you would see are these like dark brown, blackish spots that tend to start along leaf veins, because that's where the moisture tends to sit a little bit more. And then on the fruits, if you have like an apple tree, not just a crab apple tree, if you have an apple tree that you're trying to produce fruit, you can get similar looking spots on the fruit. These fruits are still edible. You can just cut out those spots. And in bad years like this year, where we had a lot of early spring rain, you can have early defoliation. And I realized I didn't have any of my own pictures, but thankfully there was a good picture on Bugwood showing how badly defoliated, especially some of the ornamental crabapple trees can be. And this was really alarming this year. People, were thinking that something more serious happened to their crabapple. But thankfully, when it comes to apple scab, it's a lot more manageable than something had we had a epidemic of fire blight, that would have been a lot more serious. With apple scab, management's pretty easy. You can start with sanitation, raking and removing fallen leaves or symptomatic fruit, because where this pathogen overwinters in those leaves, removing the leaves from the environment is going to remove that much more of the fungus and potential inoculum for next year from the environment. So getting those out of there is a really good first step. If you were thinking about planting an apple, especially like an apple in your backyard for fruit production, there is a lot of variability in host resistance. So if and you can't necessarily predict whether apple scab is going to be an issue because, it's particularly problematic this year given our environmental conditions. And our environmental conditions this spring aren't necessarily the same environmental conditions that we have every spring, right? But knowing that it can be an issue, you might consider selecting resistant cultivars to plant. And we actually have a publication or a list somewhere on our website with different apples and like their susceptibility or resistance to different plant diseases. There are fungicide sprays that you can use to manage apple scab, but timing is really important. So the way this fungus works, it infects early and it takes some time before you see symptoms. But this fungus has what we call a polycyclic disease cycle where so the kind of spores that infect early in the spring are different from those that are produced later in the year. So these are the kind of spores that I see under the microscope to confirm apple scab. These are called conidia, and these are produced over and over again. So polycyclic disease cycle means that you have essentially the cycle of reinfection. So basically, once you see symptoms on your apple or crabapple tree, applying a fungicide isn't going to do anything because you're already in that polycyclic disease cycle and you're never going to get a foothold on it. That's why timing is so important for apple scab. And this is something that like apple orchards and whatnot pay attention to for fungicide applications and make sure they get the timing right to make sure that they stay ahead of this issue before it comes a defoliating issue. The next one I'm going to go over is I have this as cedar blank rust. Uh, there are a bunch of species of gymnosporangium that cause rust on. There's one that causes rust on apple. There's actually a couple different ones that cause rust on apple, pear, and hawthorn. A little bit more about these on the next slide. This one I voted winner of coolest fungal disease because it really is like one of the coolest diseases out there. And this is one that I try to preach like maybe a little bit of tolerance for just like how cool this disease is. And I'll tell you why I think it could be a little bit more tolerated. Um, so this is another fungal disease. Cedar and juniper hosts are the place where this fungus overwinters. So this picture here is showing a gall, and these galls without the orange teleal horns are kind of like reddish burgundy, and you'll find them on the twigs of cedar and juniper trees. And in the spring, under the right conditions, you'll have the formation of these teleal horns, which produce spores that go and infect the rosaceous host, rosaceous pear, apple, hawthorn. You can see it on quince too. So the reason I think that this disease could be a little more tolerated is because you don't have a polycyclic disease cycle on the rosaceous host. Like once you see the symptoms you see are the symptoms you're going to get, it's not going to get worse. So at least there's that. But when you do have bad enough infections or in like really good, like favorable years for the spores to form on the cedar juniper host, you can see a lot of these spots form on the leaves. You can see these also form on the fruits. And these are the structures that you would see on the fruit. So this would be the symptoms on the top side of the leaf, where you would have these yellow spots with like a reddish orange center. And if you looked close enough, there would be like some tiny little black dots in there. And then on the underside of the leaf or on the fruit, you would have these kind of finger-like projections that stick out or out from the fruit. And these are the spores that are produced that go and infect the cedar juniper host. So managing this disease, one of the best things you can do again, and it's a little bit of a broken record when it comes to management for some of these fungal diseases, but sanitation is number one. And but sanitation in this disease looks a little different than sanitation for apple scab, because in this case, sanitation is going to be trying to, or like if you have a cedar tree that has galls, removing those galls. If you have a neighbor that has cedar trees with galls, you might politely ask them to prune out the galls. Um, this is another one where there's varying levels of resistance in cultivars. And this, you can find that information. We've got some publications with that information as well. And again, fungicide spray is an option, but it has to be super timely. There's a pretty long, what we call latent period between when the spores from the cedar juniper host land on the rosaceous host. Um, and once symptoms show up on our apple or pear tree, there's nothing to be done to control this. It is what it's going to be for the year. And this is again where I try to just preach, like, maybe we can just appreciate how cool nature is, that like you can have some kind of cool things like this happen. And I know that's not like a popular opinion, and I'm super biased as a plant pathologist, but this is a really, really cool disease and could be used as like a learning opportunity for kids or something. The next one, this was a big issue this year, not only in Iowa, but all over the Midwest, was aster yellows. This was seen all over Iowa in 2024. So aster yellows is caused by an organism that's called a phytoplasma. It's Candidatus phytoplasma asteris. Phytoplasma is basically a bacteria that is lacking a cell wall. They are fastidious, and that means that they live in the xylem, and that makes them particularly difficult pathogens to work with. So these pathogens can't be cultured. In order to detect aster yellows, a molecular assay needs to be done. And this phytoplasma is vectored by a migratory leafhopper. And Zach and I did an article in the Yard and Garden newsletter, where we wrote an article with our extension climatologist and have, like, maybe a guess with like data about the weather, like maybe why this was such an issue this year. We'll never be able to prove it, but the weather trends did seem to like possibly favor more movement of this leaf hopper up from the south. And one of the real stinkers about aster yellows is this is a phytoplasma that can affect over 200 host plants. So we saw it a lot in perennial gardens. We saw it a lot on purple coneflower. You get these really funky growths where the flower heads start growing like weird vegetative tissue instead of flower tissue. Here it is on Rudbeckia. So we see it in perennial crops, or plants, but it's also an issue for a lot of vegetable crops. And we've actually detected it on carrot, where it's a known issue, and garlic in Iowa this year. So that's, just like adding insult to injury with the aster yellows epidemic we had this year. And unfortunately, for managing phytoplasma, it's much like a virus disease, where in a perennial plant, once a plant has aster yellows phytoplasma, there's no cure. This is something where sanitation is really the best way forward, especially again if you detect it in a perennial garden. And the entire plant needs to be removed, roots and all, because any of that plant tissue that's still living can serve as a reservoir for the phytoplasma. And that's where weed management can be an important management method for aster yellows as well, because the weed host can serve as a reservoir for this phytoplasma. Next, this was the big, big, big thing this fall. It seemed like all of our lilacs were dying, but they weren't really dying. They were just really sad. because of Pseudocercospora leaf spot. So this is a disease that affects lilacs. Boy, did our lilacs look sad this year. You would have a range of symptoms. When it started, you'd have like these kind of irregularly shaped brown spots on the leaf, pretty like randomly distributed on the leaf. Two, kind of clustered, like over here. And then as the symptoms progress, you would have just complete death of the leaf. And then you would have, that move its way throughout the lilac canopy. And then we had really premature defoliation of our lilacs. This is another fungal disease that overwinters on fallen leaves. So another time where I'm going to mention sanitation, raking and removing fallen leaves is an important management method for this. It did seem that there was some variation in different cultivars of lilacs and how they reacted to this. It seemed like some were less susceptible. Unfortunately, we don't have a list, to document that, but that was something that I noticed this year. And fungicide sprays are again an option, and again, timing is important. So this fungus, just to give you an idea of what I see in the clinic here, when I look at it under the dissecting microscope, this is what I would see. These kind of white strands here are what's called pycnidial ooze. It's basically all of that stuff that's oozing out are spores. So once this pathogen is on your lilac, it's going to produce more spores. And this is what the spores look like under the compound microscope. So it's going to keep producing spores and it's going to keep causing new points of infection within your shrub. So fungicide sprays, once the disease has shown up, is again not really something that is going to be helpful. The fungicide sprays are not curative. They need to be proactive and like a protectant. But generally, again, our lilacs are pretty tough plants. I did hear some stories of people cutting back their lilacs because they thought they were dead. And that made me sad because now the people who cut back their lilacs aren't going to see flowering on their lilacs for a long time. While these plants look dead, they really aren't, and they should be just fine next year. Especially if you practice good sanitation. The next one I'm going to talk about is bur oak blight. I saw this a lot this year on bur oaks. This is another fungal disease called Tabacia iowensis because it was first characterized in Iowa. This pathogen overwinters on petioles that remain attached to the trees. And there does seem to be some natural variability in susceptibility of bur oaks to this disease, which is good news. But if you have repeated years of infection, this defoliation is going to accumulate stress on the tree and make it more susceptible to other issues. So in like, severe cases, you might see something like this where you start seeing like tip dieback and defoliation. This is an example of what the symptoms look like on the leaves. You typically see this browning kind of appearing in a wedge shape or along like the leaf veins like here. And then this right here is what I see when I look at this under the compound microscope. These are the fungal fruiting bodies that are producing more spores to cause to create new infection points on the leaves and throughout the canopy. This is another one where sanitation can be helpful. Not only does this pathogen overwinter on petioles that remain attached to the tree, but it can overwinter in the fallen leaves. So any amount of removing any possible inoculum for years to come is going to help minimize the amount of disease that there is, even if you can't get to these petioles that remain attached in the canopy. There are fungicide injections And again, timing is important. Um, I have been sending to clients this year where we confirm bur oak blight. Dr. Tom Harrington, who is now retired from our department, he did a lot of work with this disease, and he has a really nice webinar that he's recorded as like a half hour or so about chemicals and the timing and how it affects the outcome of the trees. So if you're interested, I'm happy to send that to you. That is an option, but again, there's also this just like natural variability and susceptibility to the trees. So it's hard to predict whether this is going to be a perennial problem once it shows up in your tree or not. And the last one I'm going to talk about is oak wilt. This is a perennial concern for oaks. And we did recover it more in 2024 than we did in 2023. So this is another fungal disease. This one affects the vascular tissue of our oaks. This is Brett-Ziella fagacearum. This pathogen is going to cause more issues for oaks in the red oak group than oaks in the white oak group. So we recovered it from both red oaks and pin oaks this year, and we have not recovered it from white oaks. And that has to do with so white oaks are more like tolerable, or they tolerate it more, like are because of the biology of white oaks, they're better at like stopping the spread of the pathogen. Um, but that doesn't mean that white oaks don't get oak wilt. It just means that it is more difficult to detect in white oaks. And then if white oaks do get oak wilt, it does generally take them a longer time to show symptoms or succumb to the fungus. This fungus is transmitted via sap beetles, and that's why we talk about pruning for oaks, especially red oaks, should generally only take place between November and February. I was on the radio last Friday and talked about this. This November's been a little warm, so I would hesitate to prune red oaks just yet. I would like it to stay colder for longer before you're pruning a red oak. But pruning when it's cold is best because the beetles aren't active, and these beetles are super attracted to wounds, including pruning cuts on the oak trees. So they will very quickly move from these fungal mats that are produced on the tree and carry spores to fresh wound cuts. Um, so what you're looking at here, this is an example of Brett-Ziella fagacearum when we successfully isolate it from plant tissue in the lab. Um, it has a distinct cultural morphology and also a distinct smell, but that's not enough to confirm that this is the fungus. So we also look at it under the compound microscope, and this is what those spores like. They look like strings of Tic Tacs. Another important management tool for oak wilt, especially if you're dealing with oak wilt in a forested setting, is disrupting root grafts or girdling and applying fungicide, or not fungicides, herbicides to infected trees to try and stop infection centers. And I forgot to mention this picture over here is showing we see vascular streaking in the sapwood when trees are dealing with a vascular issue such as oak wilt, and this is an example of what that vascular streaking looks like. Now I'm going to hand it off to Zach.
Okay, so I'm going to cover some of the insect trends this year. I have to say that the insects this year were relatively boring overall. Not really many things that I didn't expect, not really anything odd, so to speak. So I'm just going to cover the things that I saw way more than any other insect issue in 2024. The first one was earwigs. I think it was definitely an interesting case with earwigs because a lot of individuals were dealing with them without actually knowing it until we kind of got to the bottom of it. So earwigs are generalist herbivores, and they don't really care much about what they're eating, more or less. They feed on plant tissue, decaying organic matter, pretty much any organic matter that they can get their mouths on, though, they're going to at least try it. And we were getting a lot of photos that sort of looked like the one at the bottom center of the slide here, where individuals were seeing leaf tissue that was kind of chewed out in random shapes, random sizes. Nobody could find anything on their plants. A lot of folks were seeing them on potatoes early in the season. And they were saying that, hey, I suspected they were and they never found any. So whenever I hear cases like this where an individual is having trouble finding something, but they're seeing a lot of damage, I always recommend people go out after sunset, go out at 10 p.m., 11 p.m., sometime after dark, and scout them with a flashlight. And every single time that this happened in these particular cases where we saw these plants with the jagged holes, they ended up being earwigs, and they found hundreds of them after dark. So if you ever experience anything similar to that, go out late at night with a flashlight. If you can't find any culprits during the day, you might have the answer to your problem, whether it be earwigs or something else. So these, I think, were really abundant this year, primarily due to the excess moisture that we had in spring. There was a lot more rainfall, producing a lot more organic matter that things like earwigs can feed on. In terms of management in these cases, I mean, rarely are they going to cause plant death unless the plants are really small. But certainly when there's a lot of chewing, they can reduce the photosynthetic area of the plant and certainly impact that plant's ability to photosynthesize and grow. But in many cases, you can tolerate them, especially if it's in not a production setting where you're growing something for consumption. But if you do have gardens that have plants for consumption, you can tolerate them, sorry. And you can also hand remove them or create some sort of trap. So you can use traps like this where you can fill the traps with things like juice from a tuna can, things of that nature. Put it in the ground. It'll attract a bunch of them. You can take corrugated cardboard, fold it up into a bunch of different shapes, place them out in your garden, and then go out and collect the earwigs from those traps because they'll tend to go into all those crevices of that corrugated cardboard and things of that nature. But the first part here, I don't really recommend any sprays for earwigs. They tend to be somewhat tolerant and hard to manage with chemicals. So it's mostly just hand management, but the first thing is scouting to actually find them. And again, if you're seeing damage and you can't find any culprits during the day, go out at night with a flashlight. You can hopefully solve your problem, or at least figure out what the culprit is. The next one I have to talk about, the periodical cicadas this year. They were a huge topic earlier in the season. We heard there's this really big double emergence in 2024, and people were calling pretty much every day throughout the state and throughout got some calls from people in other states in the US too, saying, are they going to destroy everything? What do we have to worry about? Long story short, nothing bad happened, but we did have a unique dual emergence in 2024, but Iowa only really experienced one brood of cicadas. So there's many different broods of these periodical cicadas that emerge every 13 or 17 years, but those broods emerge in very specific locations throughout the eastern half of the United States. Iowa, we saw both broods, but primarily just one of them on the eastern edge of the state. The other brood emerged in the very, very southeast corner of Iowa. There were no areas in Iowa that we know of, at least, where both of these broods emerged simultaneously in the same location. So they were around early summer, sort of maybe creeping into midsummer a little bit, but pretty much early summer presence. And really, plants are fine. We see some flagging damage from cicadas during these really big emergences, which is what we're seeing in the bottom right photo, where the tips of branches are experiencing some dieback from essentially the egg laying of those cicadas. They'll use a saw-like ovipositor to saw into the tips of branches and deposit their eggs in there. But this is not going to kill those well-established trees. They're going to be fine. They're going to grow out of it the following season. So it's just going to be a temporary weird look to those trees, and they'll be perfectly fine. If you have saplings, or not really saplings, they're a little bit too small, but if you have smaller trees, ones that aren't as well established in the landscape, those can experience a little bit more dieback, and you can cover your plants in netting. But the good news here is we won't have to deal with these for another 13 or 17 years. But I bring it up nonetheless because it was a really big story in 2024. Next, we got a lot of phone calls about mosquitoes this year in the diagnostic clinic. So we're calling them the top itch of 2024. And this is primarily due to a lot of that early season, early summer flooding and additional rainfall. Mosquitoes require those water sources, that is mostly stagnant water, standing water sources to reproduce. So if we have more rainfall, more flooding, we can expect more mosquitoes. And I just put this chart up here that is from the Iowa Mosquito Surveillance Program at Iowa State. And we have the population of mosquitoes, the weekly average of trap catches that the program puts on in blue from 2023 and then in red from 2024. You can kind of see the clear difference. It looks like some of that photo essentially weeks out of the year. We can see that at that one point in the year where we were getting that really excessive rainfall and flooding, trap numbers were absolutely massive. So nothing really that we can do for mosquitoes other than put on protective clothing and avoid going outside in periods of high abundance. But I share it nonetheless because it was a really big trend for 2024. Most mosquito control is done by mosquito control districts through cities or county level governments. And then finally, another really big issue that we tend to see most years in Iowa, but I got a lot of news requests or news interview requests this year for minute pirate bugs, which in the media are often called no-see-ums, which is why we posed the question, did you see them in 2024? I'll briefly talk about the name and why no-see-ums isn't really the correct term for these. But minute pirate bugs are actually beneficial insects in the landscape. We see a photo here of one feeding on a thrips. Thrips are plant feeding insects that are often quite harmful pests in some landscapes. They love feeding on things like thrips, aphids, other things that are plant feeding and can be pestiferous. But what we experience in the fall is that after crops are harvested, the minute pirate bugs that are very, very abundant in those agricultural landscapes leave the agricultural landscape because there's no more habitat there after harvest. That just so happens to be the places where we tend to experience them, in our yards, near our homes, getting into our homes, etc. And then they can bite, and they have remarkably painful bites for such a tiny insect. I mean, these are nearly microscopic. That's why some people call them no-see-ums. So they are harmless. They're not injecting you with venom. They're not really doing anything but biting by accident, conceivably thinking that you're food or maybe a water source. But they are harmful. And unfortunately, management is to tolerate them. There's so many of them in the landscape that spraying isn't going to do any good because more of them are just going to come into the landscape. And once we get a couple of hard frosts, they should disappear completely. Already in Ames, we were seeing thousands of these. I went to lunch and sat outside and was getting bit by a bunch of them. And after one hard frost, most of them disappeared. But another really big trend for 2024, but one that we tend to experience every year in Iowa. Nothing really very interesting on the insect side this year. I would say the only interesting thing that I saw were Kermes scale moth, and I'm just going to put a link in the chat. This is just for interest's sake because they're a really pretty moth. And these are parasites of Kermes scale, and Kermes scale are a very common pest on oaks. So they're just really pretty, orange and black colored. I didn't even know that they existed before they were found in the clinic, in a clinic sample. But other than that, nothing really very interesting from the insect side, which is, I guess, really good for others and kind of boring for me. So with that, we left about 10 minutes for questions, which is almost perfect. So we are going to start taking any questions that you have. We'd certainly like to thank our funding organizations, Iowa State University, National Plant Diagnostic Network, and the Integrated Pest Management Program here at Iowa State University.
Well, that's all we've got. Thank you all for attending. We'll probably be sending out an evaluation after this, so keep your eyes peeled for that, and we'd appreciate if you take the time to fill that out.